Hello guys, in last video we discussed about blood introduction. Now we will see deep about RBC. Erythrocyte, it is a RBC that contains hemoglobin which carries oxygen to the lungs and tissue. RBC contains large amount of carbonic anhydrase which catalyze the reversible reaction between carbon dioxide and water, and carbon dioxide in the form of bicarbonate. Now let's see shapes. The mammalian RBC are usually non-nucleated and non-motile cells. Biconcave circular disc with central pale spot. It is shapes difference in various species of animals. Dog, cow and sheep, it is markedly biconcave. Horse and cat, it is swallowing concave. Goat, it is very swallowing or flat. Camels and deer, it is elliptical and sickle shaped. In cold-blooded animals like amphibians and birds, the shapes are elliptical and they are nucleated. Now let's see about size of RBC. The average diameter of RBC ranges from 4.1 to 7.5 micrometers. Species wise, it is goat, it is 4.1, sheep, it is 5.0, cattle, it is 5.6, horse, it is 5.6, pig, it is 6.2, cat, it is 6.5, dog, it is 7.3, and man, it is 7.5. The main thing is, and smallest and largest ones are important. Mainly goat is 4.1 and man is 7.5. And cattle and horse are approximately equal. Then now see composition of erythrocyte. Erythrocytes basically contains water around 60 to 72% and 35% are solids. Of solids, 95% is hemoglobin and remaining 5% of the cells and stromal proteins, lipids, phospholipids, cholesterol, esters, fats, vitamins, etc. Now let's see energy required to RBC. To maintain ferrous state, mitochondria is absent in mature erythrocytes. These cells can derive energy in the form of glucose metabolism via anaerobic EMP pathway, and 10% energy comes from the pentose cycle, which produce NADH and NADPH. Krebs cycle is very much reduced in the activity. Now let's see the abnormalities of RBC. RBC abnormalities mainly divided into two types: polycythemia and oligocythemia. Polycythemia is erythrocyte number increases. In oligocythemia, erythrocyte number is decreases. Polycythemia is of two types. One is physiological, one is pathological. In physiological, due to high altitude and muscular activity increases. In pathological, due to reduced in the O2 supply, bone marrow cancer. Oligocythemia is also of two types. One is physiological, one is pathological. In physiological, it is hemodilation in pregnancy, and in pathological, it is anemia. Anemia is divided into three types based on the defective formation. It is sickle cell anemia and thalassemia. And defective due to aplastic and anemic. In aplastic, functional bone marrow is not functioning properly, and anemic, megablastic, microcytic, macrocytic, and pernicious anemia. Pernicious anemia is due to lack of intrinsic factor. Macrocytic is due to lack of extrinsic factor. Microcytic is due to lack of iron, and megablastic is deficiency of iron, folic acid, and vitamin B12. Now let's see excessive blood loss. It is one type of anemia. In this, hemorrhagic accident and peptic ulcers causes, and hemolytic acute destruction of RBC. Now let's see about erythrocytes indices. Helps to find types of anemia. Mean corpuscular volume. It expresses the average cell size of the erythrocyte. Mean corpuscular hemoglobin. It gives the average weight of hemoglobin present in the erythrocyte. Mean corpuscular hemoglobin concentration, MCHC. It is the average percent of the mean corpuscular volume that the hemoglobin occupies. Now let's see about concentration of RBC in domestic animals. It is millions per millimeter cube of blood. Species wise, in fowl it is 3.0, in pig it is 6.5, dog it is 6.8, sheep it is 12.0, cattle it is 7.5, goat it is 13.0, Horse it is 6.5, cat it is 7.5, man it is 5.4, woman it is 4.8. Now let's see lifespan of erythrocyte in different species. Cattle it is 125 to 150 days, sheep it is 140 to 150 days, goat it is 125 to 150 days, horse it is 140 to 150 days, dog it is 100 to 120 days, cat it is 70 to 80 days, pig 51 to 79 days, in poultry it is 20 to 30 days. Now let's see about fate of erythrocytes. After completion of lifespan, then they will go hemolysis, intravascular hemolysis and extravascular hemolysis. About 10% it is intravascular hemolysis and about 90% it is extravascular hemolysis by MPS system. And hemolysis caused by external agents like blood parasites, basophils, rheocells, etc. and chemicals like copper, lead, nitrite, nitrate poisoning. And thank you and subscribe for more veterinary related topics.